Okay, let's start. Good morning, afternoon, evening. Hola, hello, bonjour, hola. And ni hao, konnichiwa, 안녕하세요. And hello all. And welcome and thank you, SynBYSS people, for joining us, the 100th seminar, and I call it University of Texas Austin Day. Often, numbers make me think, why do we have a decimal system, not a binary or duodecimal system? 1 year has 12 moon cycles. 1 day is divided into 12 hours. If we had 12 fingers, would we have a duodecimal system? Since the first seminar on Thursday, August 26, 2021, it has been, interestingly, 777 days or jackpot days, which would be another interesting coincidence to me. I wanted to thank all speakers and audience who support this seminar series until today, the 100th seminar. Well, every day is, in fact, a special day because we live at least one more day. Let's enjoy every second of our life because today is the day when a person who died yesterday really wanted to live. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Andy Ellington. He does not need any introduction because we all know him. He is a professor at the University of Texas, Austin. He and I meet virtually almost every month to discuss biosecurity and biosafety issues in synthetic biology as EBRC members. I met him for the first time in 2009 when I was visiting Professor Georgi Georgiou for my postdoc interview. I cannot forget that morning when he asked me, what is synthetic biology? Because I have been trying to answer that question since that day. Eventually, I chose Chris Boyd over Professor Georgiou, but Jeff Tabor and this advisee became my office mate in the Voight lab to learn more about Andy. Since that day, I have learned a lot from his insightful questions and comments on many, many different topics, and all of us do so. Andy, thank you so much for your innovative ideas and contribution to the scientific society. I'm so excited to hear again your perspectives and wisdom today. And I try to introduce you briefly, but I failed to do so. The virtual podium is all yours now. Thank you again, Andy.
Thank you very much, Dr. Moon. So I'm going to talk very briefly about a subject that I'm probably very ill-equipped to talk about, and that subject is community. And I think it's an important subject, especially today, and I'll talk about it very briefly in two ways. First is, in part, as an introduction to Dr. Adamala, who I know Dr. Moon will introduce in just a moment as well. But community is about who we are. And I think, in particular, Dr. Adamala and I are both alumni of Jack Szostak's lab, and so we sort of know each other at arm's length in that way. And we brought along with us into our own academic lives certain pieces of that culture. But one piece that I think is really important that I think we did learn in the Szostak lab and elsewhere is that in community, you take care of each other. And so in this regard, my introduction of Dr. Adamala has to do with an email I received from her yesterday, where she writes to me and says, "Hey, Andy, there's a student from Belgium who would like to come to this conference that I'm hosting." It's actually the Build a Cell conference, and Kate is huge in the cell-free, build-a-cell world. She's essentially built that entire structure from the ground up. And so it's amazing, and she'll probably talk about that herself. This student received a travel grant, and I'm giving her a maximum amount because she's not US based. I'm biased, but I'd like to go out of my way to help her because she's from a small Polish town not far from where I grew up. I managed my first summer in the Shostak lab only because lab members were helping me couch surf. So again, it's interesting to me because, again, I think the world of Dr. Adamala, but she demonstrates how community is helping one another out. And what she doesn't realize is that when I was a graduate student, that there was a point, now this was before I was in the Shostak lab, there was a point where I just really wanted to go to a Cold Spring Harbor conference. But I was a very young graduate student, and nobody's going to send me to a Cold Spring Harbor conference. And so I know this is very hard to imagine. I just got on my motorcycle and I rode from Harvard out to Cold Spring Harbor and said, I'm at the conference, I'm attending. I didn't enroll, I didn't badge up, I just showed up. But fortunately, there were other students there who, when the conference organizers inevitably found out who I was and tried to kick me out, stood up for me and took care of me. And so when I was a graduate student, the only way I was able to do a lot of things, and that's just one of them, was because people took care of me, because I had a community. And I try and carry that on till today, just as Kate does, just as I'm sure many Shostak Lab members do. The reason community is so important, and the reason that we need to take care of one another, should be manifest from various world situations today. But Tesak and I and Kate are all part of the so-called security working group for the Engineering Biology Research Consortium, the EBRC, where we discuss a variety of security issues. But for me, all security issues that we discuss come down to one thing in synthetic biology, and that is we need more white hats than black hats. We need more good people than bad people. And so whoever's listening to this in the synthetic biology community, we need to take care of one another. And in particular, we need to just all work together as best we can as white hats so that our community can flourish and so the world can flourish. Even in these obviously disruptive and dangerous times, we all need to stick together and be a community. And with that, I probably said way too much for way too long, and I'm going to turn it over to my peers who will say much sager and more meaningful things than that. Thank you very much.
Oh my God, Andy, I wanted to hear your talk next 1 hour, but of course I need to do the time to. I mean, you always give us so meaningful, insightful things, and I wanted to think about that point next, 1 month. I mean, that's absolutely brilliant talk, and I 100% agree with you. Small thing we share with other people that makes the community, that makes this world much better place to live. And then, so absolutely wonderful. Thank you so much. And that's amazing. All right, so thank you. Now, the main speaker of today with a little bit longer introduction, Professor Kate Adamala is McLeod Land Grant Assistant Professor at University of Minnesota. I mean, based on what she achieved, I still don't believe she's still assistant professor. She should be, I mean, full professor by now, but she's still, assistant professor. She is a biochemist by training, running a lab working on engineering synthetic minimal cells. Importantly, as Andy already mentioned, Kate is the fearless leader of the EBRC Biosecurity Working Group, where Andy and I discuss multiple topics on biosecurity and biosafety. She is also, very importantly, leading an international synthetic cell engineering community. That is what Andy already emphasized, called Build a Cell, and co-founded synthetic cell therapeutics startup SynLife. Additionally, she has run the Build a Cell weekly seminar series for many years, demonstrating her true leadership in synthetic cell research. As Andy already mentioned, interestingly, both Andy and Kate are alums of Professor Jack Szostak at Harvard, of course, also a Nobel Prize winner. Hopefully, both of today's speakers will win Nobel Prize in the future. Kate, thank you so much for your time today and also your amazing leadership. And I believe you are the true rising star or pioneer already. And also thank you for your amazing leadership for the community. Again, I want to emphasize the word community. And please take it away, and all yours now. Thank you.
Thank you so much. Thank you, Tae-Suk, and thank you, Andy, for that introduction. Please mention that to my tenure committee. My package is about to go in, so any endorsement is appreciated.
No, of course. I mean, I wanted to talk to all the professor who potentially writing letter. You should write the strongest letter. Period. Thank you.
Thank you so much. So you should be seeing my slides right now. Thank you. So thank you so much for having me here. And what I want to talk about today is I really appreciate the introduction that emphasizes the community. This is a very collaborative field. This is the field that solves problems that cannot be solved by a single lab or even a single country. And this is something that drew me to this field, this real sense of collaboration, cooperation, rather than the competition. And I will talk about that towards the end of my talk. But I want to spend the first maybe 20 minutes talking about the science itself. Why are we doing it, and what is that we're doing it? Why are we doing synthetic cell research when we have perfectly good, normal, natural biological cells? This is a valid question. Most people that have ever done any biological research are probably used to thinking about biology as this incredibly diverse, varied field. And that's true. There is a lot of form and function in biology. If you look at the modern tree of life, there is so much. There is so much variety in function, in form, that you really might think that nature has done it all and there is no need to study anything else. We can just spend our entire life studying just the natural biology and we can learn everything there is to know about life. Unfortunately, from the chemical point of view, I was trained as an organic chemist, so to me, I'm thinking of life as a biochemical event. And from the chemical point of view, life is actually pretty boring. When you think about it, all of life uses the same 22 amino acids. All of life uses the same 5 nucleobases. All of life relies on a central dogma where DNA, RNA, protein-based biochemical processes are identical across all of the domains of life. And most importantly, all of life comes from the last universal common ancestor. So all of life comes from this one source, and therefore there's not that much biochemical diversity in life. And so this is the questions that our field is asking. What else is out there? What can we do, even with the same building blocks, the same types of building blocks, but what kinds of forms and functions can we investigate? And what are other ways in which nature can do life? So life as a physical chemical phenomena doesn't have to be the life as we know it right now on Earth. Life can be a lot of different things. And most importantly, we don't really know what is that? What other kind of possibilities are there for biology? And even more importantly, we don't really know that much about our own life. When we think about cell, we think about biology that the business model of biology is to try to investigate a modern living cell and try to control it. We don't know how to do it. That's one of the things that biology as a field still unfortunately failed to deliver, is how does a modern cell work? How does a cell as we know it, how does this complex biological assembly work? And that's one of the things that bioengineering has been talking about for ages now, since the origin of that field. And that was, how can we kind of investigate life? How can we investigate cell? And how can we look at a modern cell and figure out exactly how it works, exactly where every molecule goes? And we still can't. Even though we have a cell with a completely artificial genome, the JCVI-based mycoplasma synthetic cell, we still don't know a few dozen genes in that cell. We still don't have a full chemical map, ingredient list of any living cell. And so that's what my field is trying to do. We want to build a cell that's simpler than a complex living cell. And we want to build it from known components so we can truly understand it. There is a quote from Richard Feynman that's heavily overused in our field, but I think it's still very relevant. He said, "What I cannot build, I cannot understand." And that makes perfect sense to me. If you cannot build something from scratch, you cannot really say you understand it. And so that applies to every process in biology, in engineering, including life. If we cannot recreate life, the process of origin of life, we cannot really say we understand life. So how do we do that? There are two ways in which you can approach that problem. This so-called top-down and bottom-up approaches. So top-down approach says, we already have life. We have living cells. Why don't we try to minimize it? Why don't we try to take away elements until we arrive at the simplest possible system that's still alive? And that will be our simplest minimal cell. Now, the bottom-up people say, let's start with something we know, and that is purified components, very well-defined components. And let's start putting them together and put it together into an increasingly complex system until we arrive at something that we can finally say this is a cell. Both of those approaches have advantages and disadvantages. The bottom-up approach have not resulted in making a fully living cell yet. We're putting together components into something that looks like a cell, smells like a cell, starts to quack like a cell, but is not yet a living cell. The top-down people made something that's definitely alive, it self-replicates, it has a cell cycle, but is not fully defined. So neither of those approaches really resulted in a fully living, understandable, controllable synthetic cell, but both of those approaches are complementary. That's where the field is right now. We're at this very fascinating time where both of those approaches are about to meet in the middle, and we are about to bring in this so-called synthetic cell, a fully controllable synthetic living organism. And why are we doing it? What would we do with it once we have it? Well, one of the most interesting things about it is the modularity of the synthetic cell. Whether you use the top-down or a bottom-up approach, the synthetic cell will combine elements of whatever you want. In our case, as biological engineers, we want to combine elements of different living systems that we know of, so different genes, different biochemical pathways, into recreating existing pathways, but also building new pathways, building new ways of doing life, ways that biology have not invented yet. So when I say a synthetic cell, there are many different ways in which you can define that organism. I define it through the practical approach. I'm a pipetting monkey. I think about everything in the context of whether I can actually do it in my lab. So when I say a synthetic cell, I mean a liposomal bioreactor that expresses proteins. It's a vesicle, a lipid vesicle that encapsulates a cytoplasm made from a cell-free protein expression system. And in the membrane of that cell, there are channels that allow us communication with the environment. Our genome is most of the time a DNA genome. Sometimes we're moving away from DNA. We're entering that kind of crazy science fiction realm of building things from scratch that don't exist in nature. But right now, most of the time, we focus on the DNA genome for practical reasons. There's no easy way of defining synthetic cell. This is kind of a functional definition that I use in my lab, is that soap bubble, a lipid vesicle that expresses proteins. I'm often pressed to give a definition, to have a more general definition of a synthetic cell. So this is my general definition of a synthetic cell. It's something that has not been alive before the experiment started. So if you think about it, all of the life forms that exist right now on Earth come from an unbroken line of life since the last universal common ancestor of all organisms. So there's never been, after the first origin of life event, there's never been another origins event that made it till today. There probably were plenty of origins events, but they didn't make it. We are descendants of that one single origins event, and everything that lives right now is descendant of that one origins event. Synthetic cell research aims at making cells that are not descendants of that one origins event. So basically making life from scratch. And the really important thing is we call ourselves bioengineers. So we really want to be able to control our system. So a synthetic cell has to be more than the sum of its parts. It has to have some emergent properties, because otherwise it's just a biochemical experiment. But it has to still be controllable. So as we're building up complexity to build more and more complex synthetic cells, we cannot lose the sight of that controllability. And some things that we can control in synthetic cells that are impossible to control in living cells are things like the size of the cell, the composition of its membrane. You cannot take an E. coli and blow it up by order of magnitude and expect it to still be alive. But you can imagine doing that with a synthetic cell. So we can also control the genetic systems that run the cell. They don't have to be DNA. We've used those so-called xenonucleic acids, so artificially made nucleic acids that have either different backbone or different nucleobases or both. We can mess with the translation system. We can do things that nature has not bothered doing in 4 billion years. For example, we are right now evolving the catalytic center of the ribosome, the peptidyltransferase center, that's one of the most conserved sequences in nature. We can evolve that in a synthetic cell. We can also evolve things like the codon table. Most of the life uses the same codon table, and the organisms that have some variation usually have variations in just few codons. The kind of a basic skeleton of the codon table is unchanged. We can change that. We can reprogram pretty much every codon in vitro in a synthetic cell. So we can change every aspect of every fundamental aspect of the cell. We can change every aspect of the central metabolism. We can change the whole design of how the metabolism runs. So how do we do it practically? So I showed you this as this is what I call a synthetic cell in my lab. So I'll keep that as a blueprint. I'm obviously biased. That's what we use in my lab. And so that's what I want to keep using. So my synthetic cell has a membrane. I think every honest cell should have a membrane for evolutionary purposes to maintain the individuality, but also for practical purposes. You want to keep the guts of your cell together. So most of the time when we have a membrane, we have a lipid bilayer membrane. So our cells have a membrane that's not dissimilar from modern biological membranes right now. We use phospholipids and cholesterol to build very stable fluid membranes that are resistant to most environmental factors. They're easy to make. Sometimes when we do origin of life experiments, when we try to kind of reconstitute those earliest evolution periods, we use fatty acids. But for most experiments, we do use honest, well-behaved membranes made of phospholipids. Sometimes we don't use lipids. There is this giant field of droplet microfluidics. Here's a video from my postdoc Theo, who is a microfluidics expert, and he was using mostly microfluidic systems to build those very uniform droplets. Those droplets don't have a membrane, but they encapsulate the guts of the cell, so the cell-free protein expression system, in a water-oil emulsions. Now, regardless of what kind of encapsulation system you use, the really important part is what's inside. And what's inside our synthetic cell is a cytoplasm that's made right now mostly of natural components. That's mostly for practical reasons. We do not have an artificial ribosome system yet that we can use. We do not have a robust artificial genetic system. So most of the time we are using natural systems. A cell-free protein expression systems mostly derived from E. coli, because that's the cheapest and most robust way. But you can also derive those systems from pretty much any domain of life, all the way up to the human cell lines. And so what is a cell-free protein expression system? It's literally guts of a cell that were purified and broken down to its elemental components. So we purify every protein separately and put it back together with all the small molecules. So we maintain this chemical definition. We know exactly what goes into our cell-free protein expression system, and we can design it and we can control it any way we want. So we can basically design those systems to either express the natural proteins or express unnatural proteins with unnatural amino acids. This is the chassis. It's like a blank chassis that we can engineer on. So what do we do with it? This field is defined by applications. It is essentially a foundational research problem. How do you build cells from scratch? But we're doing it for a reason. We're not just doing it because we think it's cool. I mean, it is inherently cool, but we are doing it to solve real practical problems. So some of the problems that my lab is investigating are, some of them are those kind of a foundational research problems. So understanding natural biology. We're building synthetic cells so we can understand natural cells. We're hoping that if we can reconstitute natural processes in a synthetic cell system, then we can study them literally to death. We can study them beyond the point in which you cannot study them in the natural system because natural cell, for example, gets dead if you dysregulate it too much. In synthetic cells, because they're already dead, they cannot get any deader. We can study some of those processes, like, for example, translation, speed of translation, post-translation modifications, transcription, speed of transcription, folding of mRNA, all those processes that are really difficult to study in a natural context. We are also building tools. One of the most fascinating aspects of this field is this interplay between tool development for engineering of synthetic cells and use of those tools in other fields. For example, any of you who've ever done any biological engineering probably are familiar with the Gibson ligation. It's one of the most common cloning techniques used these days. And that technique was developed by Dan Gibson during his time at the Craig-Venter Institute when he was working on assembling the genome of the first mycoplasma-based synthetic cell. He was faced with the problem of how do you assemble this giant single chromosome of a synthetic mycoplasma cell? And in order to do that, he invented what is now called Gibson ligation. And that technique spread all through the biological engineering and is now a very common cloning technique. So this shows you the tools that we make to build artificial cell systems are also finding use in other areas of biology. Then other foundational research questions are around astrobiology and origin of life. How do you study those earliest evolution of life, those processes of the earliest evolution of life? How do you study the kind of a point when matter becomes life? You can study that within synthetic cell system. And interestingly, not only you can study that in a terrestrial context, so you can try to recreate the origin of life on Earth, but you can also try to recreate the origin of life elsewhere. For example, we as humanity, then NASA is working on preparing a sample recovery mission to Enceladus. And we're thinking about a mission to possibly collect samples from under the ice of Europa. So these are places in the solar system where there is liquid water, there is enough energy to possibly support life. But life that we're going to find there, if we find anything, is not going to look like terrestrial life. It's not going to be an E. coli bacteria waving to you. If we find E. coli there, then probably means we contaminated that already. What we're going to find is going to be a life form that's going to look nothing like terrestrial life. And we don't know what it's going to look like without the ability to truly recreate those origin processes under those conditions. So that's what we're doing, is we're recreating origin of life processes, not only under terrestrial conditions, but also under conditions that could possibly be present elsewhere in the solar system, including early Mars, including early Venus, current Enceladus, current Europa. How could life originate in those places, and how could that life look like? And then practical applications. Things like diagnostics and therapy. I'll show you in a second how synthetic cells are already finding practical uses in the biomedical space. We're using synthetic cell systems and cell-free systems for metabolic engineering. There is already plenty of companies that are using cell-free systems to discover metabolic engineering platforms, to build better drugs, to build better metabolic engineering pathways. We're using it for biocomputing. There's been this big promise of biocomputing that we're going to build biological computers, they're going to be awesome, and that haven't happened yet. Mostly because natural cells are not dumb. They're not going to do things that are against their best survival evolutionary interests. But synthetic cells can be programmed exactly for what you want them to do, because they don't have that underlying metabolic complexity. or evolutionary complexity, you can design them exactly to do what you want. So this kind of revived some of the promise of the field of biocomputing, that we can actually build reliable biocomputers. There's a lot of applications in biosafety and biosecurity. Synthetic cell systems are a very good, relatively safe chassis for engineering different scenarios, different synthetic biology evolutionary scenarios. Synthetic cells are also a great tool for low resource biomanufacturing, and that includes space exploration. And I'm going to show you an example in a moment. So these are some of the possible applications of this field investigated in my lab and also in plenty of other labs. And I basically spent the last, 10 minutes already talking to you about the advantages of making cells from scratch, making artificial cells from scratch. And you're probably thinking, some of you might be thinking, this is incredibly difficult, probably insane, and it will never be done. And you're not wrong to think about it. A lot of the time when science is on the edge of doing something that had never been done before, it seems impossible to people. And I want to show you a quote from one of my favorite articles in the New York Times. It's a very respectable journal. And in October of 1903, they published an article on the feasibility of flight, of building a flying machine heavier than air. And the conclusion of that article was that the flying machine will never happen. We basically might as well use our resources better, and we shouldn't be wasting time trying to build a flying machine heavier than air, because this will never happen. And 3 months later, the Wright brothers flew their first Wright flyer. So the reason I love this quote and I love showing it is because if you tell engineers that something is impossible, we're just going to go and do it. We're just going to go and prove that this is something that is absolutely doable and will get done. And so that's why I kind of love this comparison. Something that seems impossible to most people is actually going to be done if enough engineering thought and resources are applied to it. So I want to spend the next couple minutes showing you a few results that are not from my lab, but I just want to show you kind of the overview of where this field is. And then I'll show you one more project from my own lab. So I talked about diagnostics and therapy, biomedical applications. I want to show you 4 different the highlights of work from different groups that show the boundaries of applications of synthetic cell technology. So the first is, I talked about biomedical applications. This is a figure from a paper from Avi Schroeder's lab at Technion. They've demonstrated that synthetic cells can be used to shrink tumors in a living mouse. So this is a picture of a stained breast cancer tumor model in mouse, and they injected into that tumor synthetic cells that produce an anti-tumor protein with an anti-tumor activity. And they show that you can shrink that tumor, you can kill those cancer cells without affecting healthy normal cells by using those synthetic cells as extremely targeted cancer therapeutics. So this is kind of at the boundaries of the biomedical applications of this technology. Now, another paper demonstrated that you can use synthetic cells to grow vasculature in organoids. This is that the yellow on those pictures are stained blood vessels, and the growth of those blood vessels was induced by synthetic cells that produce growth hormone that stimulates the growth of those blood vessels. So synthetic cells could possibly be used in regenerative medicine. There's proof of principle that demonstrates they could be used to regenerate specific tissues. Now, cell-free systems and synthetic cells can be lyophilized and stored at room temperature for a long time. This is a figure from Jim Collins' lab with Keith Pardee as a first author, who's now a PI on his own, who works on making those portable on-demand biomanufacturing systems based on cell-free systems. You can freeze-dry the thing and lyophilize it, store it at room temperature for a long time, and then rehydrate it and make vaccines that work in vertebrates. Another example of biomedical uses is this is work from Fritz Simmel's lab, where they demonstrated that synthetic cells can be used to make bacteriophages that then can produce antibiotics, and that induce either antibiotics or the bacteriophages themselves have activity against antibiotic resistant bacteria. So they call them factories as phages that produce therapeutic molecules. And that's done completely from a cell-free system. So this shows you kind of the boundaries of it. Now let me show you very briefly a project that my own lab was involved in, and that is the use of synthetic cell systems in this extremely low resource environment. So we call this project astropharmacy because it was geared towards demonstrating that synthetic cell systems can be used to produce drugs under spaceflight conditions. This is obviously for space exploration, but if the implied benefit is that if you can do something under spaceflight conditions, you can also do it under low resource conditions on Earth. So those technologies can also be used for biomanufacturing on Earth. So I was lucky to be a part of this amazing team of engineers and scientists and pharmacologists, pharma people that work on trying to build a astropharmacy platform that uses cell-free systems and synthetic cells to create biomanufacturing system that makes functional RNA and functional proteins. So people talked about using it under space-like conditions for a long time, but no one actually demonstrated that it can work. And so that's what we did. We designed two different experiments, a transcription experiment where we produce a fluorescent RNA aptamer to show that we can produce a functional RNA, and a translation experiment where from the lyophilized cell-free system, we produce a fluorescent peptide, a small fluorescent peptide. So we lyophilized those systems in a test tube. We stored it at room temperature. We load it up on a rocket, and that rocket launches. The reason I'm showing you this video is because this was one of the coolest parts of my career. So this was really early. This was 3 years ago now. That was very early in my faculty career, and that was the first time my own sample was going into space. So that was obviously super cool for a nerd like me. And it was like, I made a sample, I put it up on a rocket, and I shot it into space. It was a sample return mission. So we did recover it, we analyzed it, and we demonstrated that, yes, you can express proteins under spaceflight conditions. Those proteins are active, and those proteins and functional nucleic acids can be produced under those very demanding low resource conditions. So now we're working on an ISS mission where the actual human astronaut will set up those experiments and monitor it in real time using the onboard plate reader. So that's kind of the limits of this technology. You have true biomedical applications, you have low resource environment possibilities, and there's a lot of types of applications that I haven't talked about for the lack of time. But one of the really important things that I do want to spend the last couple of minutes talking about is we're not only building those applications, we have to make sure that those applications are developed in a safe and secure way. So this is something that that's a big part of my work, is biosafety and biosecurity aspects of synthetic cell engineering. And some of the things that we think about is the safety frameworks. We can use the safety and security frameworks from current SynBio solutions, but there are new biosecurity needs that need to be invented for those truly lineage agnostic organisms. So some of the issues that we're looking at are things like dual use concern. We need to develop a robust malware analysis platform for those systems. We need to develop new regulatory and policy considerations for that field. They don't exist right now. And as we're growing that field, we have to be doing kind of advanced horizon scanning and thinking, how are we going to do it? And who are we doing it for? Well, obviously, we're doing it for the research community. We're doing it for people that fund it. We're talking to the regulators, the people that actually are responsible for making those laws. But we're also doing it for the public. There is a growing citizen science movement, people that do experiments in their own garages, people that work on biological engineering in their own time. And those people also need safety and security regulations, and they need chassis that they can work with that's safe and secure. And speaking of that amazing community, one of the things that really drew me into that field is how friendly it is. And as an incoming assistant professor, literally in the first year of my faculty job, I did something that's absolutely stupid and no one should ever do in the first year of their faculty job, is I got involved in organizing the synthetic cell community. So we started this so-called Build a Cell organization, which is an open collaboration for people that want to engineer synthetic cells. We are an international community. People early on recognized that building a cell from scratch is something that cannot be done by a single lab or even a single country, nor should it be done. We should work on it as international community, and we do. This is one of the best parts of the field for me, is that we are actually an international community. We have people from all over the world working with us, sharing ideas, collaborating, and we're doing it in ways that hopefully really encourage collaboration and hopefully provide a blueprint for other fields. So one of the things we do is we do not define the end goal. A lot of the time people ask, how do you know you're done? Well, I don't have a good definition of life. What we're doing is we're encouraging collaboration, we're coordinating collaboration, and let come what may. We see what people can build and at which point we decide we actually made a synthetic cell. What we do is we coordinate research. Especially we coordinate sharing unpublished data, failed results. We want to improve reproducibility. We want to share reagents. We want to share protocols. We also really emphasize interpersonal collaborations. We want people to meet with each other. We want not only the PIs, but also the students to know each other. We have workshops, in-person workshops twice a year. The next Build a Cell workshop is actually hosted by Andy in Texas, in Austin, in November of this year. You can still register. And those workshops every 6 months bring the community together. We have people that actually travel there every 6 months for 1 day to meet with other people. And that way we build that community from the ground up. People on every stage of their career, but mostly students, get to know each other. They get to have those lines of communication open to working groups and also just to personal connections. We do a lot of outreach communication with the general public. We produce materials for the public. We also do science policy work. We work on safety, security, regulation, containment issues. And shout out to the NSF. We are NSF-funded research coordination network. So I'm out of time, so I want to end by thanking everyone. Most of all, I want to thank my lab, those amazing people that joined my young lab when I started my lab and I said, I want to build life from scratch. And there were actually people that said, this is not crazy, and they want to join my lab, and they did. And I'm incredibly grateful to them. That's probably the best part of being a PI, is that I get to work with all of those amazing people. And I want to thank all the funding agencies. This would not be possible without people believing that this is something that's worth supporting. So I'm extremely grateful to all the funding agencies and to the people that work with me. I'm also extremely grateful to all the collaborators that I don't have time or space here to acknowledge, but everyone who worked with me during that time. So thank you very much. And I would be happy to take questions. And if you have any questions, if you're watching the video and you have any questions, you can just always shoot me an email. Thank you.
So Katie, amazing. I mean, I going to say two things. One of them is I am doing some research regarding sending bacteria to the moon or ISS, supported by the DARPA. So hopefully next year, you and I go to the moon together for the summer vacation. And second thing I want to mention, I want to thank you because I form collaborations, multiple actually, not only one, multiple collaborations through your speaker series. After some speaker gave a talk, I reach out to those people. I already, form a collaboration with multiple people. So that is absolutely amazing service activity to the community, and that's brilliant. And I want to say thank you one more time. So I want to give Andy, do you have any question or comment on her talk?
No, it was brilliant. It was a broad and wonderful overview. And so I would turn it over to our audience members.
Okay, so I have the one question already from audience. So Maya asked a question. Thank you. How do you encourage sharing negative result? What channel do you use and how do you document this? That's an interesting question. I have some answer, but I want Kate to answer that question first.
Thank you very much for that question. Yes. So I want to start with saying it's incredibly difficult. It's really difficult, but important to force people. We literally have to force people to do that, but it's worth doing. sharing unpublished negative results. We're doing it right now in two ways. One is through working groups. So within Build a Cell, we have working groups that work on specific problems, specific kind of areas or aspects of synthetic cell engineering. And one of the things that they do is they define very specific problems. For example, how do you make vesicles in a reproducible way? And that kind of a question is then distributed to other member labs of Build a Cell that work on those problems. And they can share not only their answers, but also their negative answers. So for example, I always have problem making vesicles using this technique, even though everyone else says it should work. My lab always has problems with it and we don't know why. And information like that makes it to our protocols. It makes it to those specific Build a Cell protocols that we then make available to everyone. And then people that encounter similar problems can say, all right, someone else already had it. So that's the relatively easy part. The more difficult part is sharing negative results and negative results of whole projects. So one of those things is, for example, someone has an idea of, let's say, how do you build a genome replication system for a synthetic cell? And they develop a very complex polymerase-based system that should work, and it doesn't work. So that obviously never gets published because it's a negative result. How do you let other people in the community know that you already tried it and they might as well not waste their time trying it? So what we do is we encourage posters that present negative results. During those Build a Cell workshops, we don't have research talks, but we do have poster sessions. And we always encourage people to do the thing that you normally don't do, which is prepare a poster on your failed experiment. Show me the project that you did during your first year of grad school when you were just screwing around and you failed. And show it to the whole community. So then people might know that you are interested in it. And they can either say, okay, then I'm not going to bother wasting my time trying to do it. Or if they have an ideas that you haven't tried, then they can actually either share the ideas with you or start on that project without needing to reproduce the mistakes you already made. So this is what we're doing within the community.
That's
and I'm
wonderful.
happy to hear what.
Yeah, that's wonderful. So that I hope that work. I want to share my experience, my personal experience, and then what I want to do in the future. So actually, I try to include negative data in the supplementary, information document when I submit my papers. First one, interestingly, reviewer asked me to remove it. So I really didn't understand why. So I remove it. And then we try again for another paper, but I'm not sure the same reviewer or different reviewer, the other reviewer also asked us to remove that data from the supplementary information. And then that happens all the time. So I don't know, that is because of the community not still accepting the concept of negative data, but I talked to many people want to see negative result. In that way, avoid any unnecessary wasteful experiment, repeat. So I absolutely, wanted to see negative result, but that's not happening. That's not happening right now, unfortunately. So one thing I want to do as editors of multiple journals, I wanted to create a special topic that is only accepting negative result containing all about the negative result, and then just submit, and then we will consider publication of those one if that negative result really giving us the insight. So I'm gonna do that. So we started discussion about that special topic in some journals at this moment. So I hope that will launch soon.
Right,
Andy,
so
you have any comment, I guess?
Yeah, I do. Based on what you just said, I think I'm more in agreement with you and Kate than I would have thought, because I'm going to answer a different question than Maya asked, which is not how do you put your negative results out there, but how do you get credit for your negative results? That may or may not be the question you're asking, but the question is, if you put in a lot of hard work, how do you get recognized for that? I would argue that you will not get recognized for negative results, period.
Mm.
And I don't care if we make a journal for it. I don't care if we have a community. It's very difficult to get recognized for negative results. But what Tasak said there at the end is important. It's the contextualization of your negative results that's important. It's your intellectual effort that shows how your negative results fit into a whole that will distinguish you. So whether you did a thing or didn't do a thing, or whether it worked or didn't work, what the community and what individuals, I think, will respect, what science will respect, is what did you bring to the table intellectually?
Mm-hmm.
So if you know how your quote unquote failure, and I understand that a negative result is not a failure, but how a negative result improves our understanding, as Tasak just said at the end there, then I think that's incredibly valuable. So right now I have a postdoc, I won't name them, who for 2 years has been attempting to do a thing, and it has failed and failed and failed. And I'll just use the word failed. It has failed. This person has lots of negative results. Now, most recently, they got something to succeed. That's great. But what it actually has made me do is contextualize the negative results. Oh, I now understand why all these other things didn't work. And I fully intend, I'm with you, Tasak, to put the negative results in the manuscript I hope we eventually write, because it's like, oh, we tried this, and the reason it didn't work was because of this. Now, again, without the positive result, could I have made the explanation of the negative result? Maybe not. But even before we got the positive result, I was trying in my own mind to contextualize why. What's going on? Why is this system working this way? Mm-hmm. And while that would not have made a great paper, it would have made a paper that would have been of greater import than just this is a negative result. So I think I'm just expanding on what you just said, and I'll stop there.
That's wonderful. So thank you. That is you explained the better what I want to say. So that's amazing. Let me check though. We have another question. If the Andy does not have any question, I want to ask some difficult question to Kate. If I understand your remark correctly, I believe you mentioned a single ancestor cell led to all other organisms. Am I right?
A single population of ancestral cells.
Oh, single population of the ancestors. So that means you think not just one single cell led to other organism, but population. But the population not necessarily in the one location, but probably different location, right?
My bet would be on a single location, and I can see Andy is to be talking to join that conversation.
So actually, I want to just throw that question to everybody, because I wanted to understand, because the evolution always make me excited, but I still don't know whether single cell origin or multiple cell origin, which one is the right one. Oh, I don't know. I mean, it doesn't matter which one is right, but that gives us some insight when it comes to do research at the end of the day. So I want to hear your perspective or opinion.
Right. So Kate is by far the better evolutionary biologist of the pair of us, but you're pushing at some really deep questions in the field, which revolve around, even from Bose, Carl Woese onwards, whether there's a consortia of genetic information that made it to the modern day, or a single origin, a single last universal common ancestor. And the fact that we all agree that there's been endosymbiosis along the way sort of leads one to believe that certainly there's been a pastiche of genetic information that's made it to the modern day. But whether the endosymbiosis events were themselves descended from a single organism, I think, Kate, I'm just going to say, is an open question. The notion that there was great horizontal exchange that led to the LUCA, led to the last universal common ancestor, I think is probably one of these questions that I would call an agnostic question. We'll never really know the answer. Now, again, people have done amazing things with sequence comparisons and reconstructions. Maybe we will know the answer. Maybe the Asgardian organisms are going to give us, new insights into the answer. But that's my opinion, Kate, is we're asking a question where the field will never have a definitive answer, and it's possible there was a LUCA, and it's possible there was a consortia. It just is very difficult to tell since we've had horizontal transfer on top of all of that.
I think that's the single thing almost everyone in the field of origins agrees with, is that we'll never know. We can make theories, but until we invent the time machine, we won't actually be able to go and pick up a sample from that environment.
Okay. So my second, another challenging question, I probably asked that question to other people, I guess. So you mentioned life is so boring, including only 20 amino acids, 4 nucleotide, blah, blah, blah. I always wondering why 20, not 19 or 21. It doesn't make a big difference, I guess. But somehow that's related to original life question or different question. But I just wanted to hear from you, because you are the expert in that, boring life.
I don't know why the particular amino acids, and there are a few people that made their entire careers out of investigating it. And the explanation that appeals to me is this frozen accident. The codon table did not emerge all at once. It was built sequentially. That's why we have a wobble base. That's why, probably the original codon was just few amino acids. It was probably still 3 letters, but the base did not matter as much. And then as their life was evolving, we started needing more chemical functionalities and more amino acids were being added. I don't know why those particular ones. I don't think anyone knows right now. Mm-hmm. There are some theories of, obviously, we need some hydrophobic, some hydrophilic. It would be good to have disulfide bond formation, all of that. But why that particular chemistry and not, for example, one more carbon in the chain or something? I don't think we have conclusive answers to that.
Right. And so I completely agree with Kate. But I think that a subset of your original question is a really interesting way to think about it, which is, why did we stop at 20 amino acids?
Yes.
Now, which 20? Kate and I can agree it's a frozen accident. They were added sequentially. This could have to do with availabilities, could have to do with simplicity of metabolic pathways. There's a whole number of ways you can get to 20. But then why didn't we go beyond 20? Because we, of course, did. We have, for example, selenocysteine as very specific 21st amino acid, pyrrolysine as a 21st amino acid. It's not as though the code is so sacrosanct that you can't expand it. Nature has expanded it, and via directed evolution and other engineering feats, we've expanded it in the modern era as well. So why did we stop at 20? What was the limitation? And most recently, in fact, in part because of the failed experiments I just talked about, I'm leaning towards it's too big a task for EFTU beyond a certain point. EFTU is one of the most amazing proteins ever. And it's doing this incredible job of balancing the loading of every amino acyl tRNA complex onto the ribosome. That is a monster task. That's why you have selenocysteine incorporation, you have cell B, you have the EFTU equivalent. And so nature decided, I can't throw things out of balance. Once the system got complex, it would have been grotesquely thrown out of balance to add a 21st. Now, maybe that would have been possible earlier on, and maybe the amazing EFTU would have said, sure, I can juggle 21 or 22 or 59 or whatever. But I think that there came a point very early on, even after we sequentially established the code, that EFTU became the limitation. tRNA is not the limitation. tRNA is amazingly flexible, but EFTU has this incredibly hard job that I think reached a limit. And so that I'm just putting out here today based on negative results, but that's my view for why we don't have more than 20, not why we have the 20 we have.
I see. That is very interesting. Thank you for sharing. I want to continue, but I see the 11:04 in my time. I have more question. Okay, so Maya asked another question. Mostly the question is because I want some kind of a database where I can make sure I'm not inventing a triangular wheel. So that is probably not gonna question. And then the last question is, what's that? Oh, does this have to do with the on-land-back Chapman basis and amino acid affinities combining to make EF2 bind properly?
Right. So what Maya is very nicely asking is whether or not this is part of the balance hypothesis. And yes, it's to the extent that EF2 is trying to balance tRNA loading by having a differential binding to the amino acid portion and the tRNA portion, there are limitations, I think, when you introduce a new amino acid as to how well you can change the surface and still achieve that balance. The fact that over multiple tRNA and amino acid pairs, you've achieved that balance is just a miracle, in my view. And so treading upon that miracle with a 21st amino acid, I think, has become too hard, which is why when systems invent a 21st amino acid, they invent specialized insertion systems for them.
I see. That is interesting. So let us close and then we probably chat offline more. So thank you all for joining and staying today, the celebration of 100th seminar. We'll meet again next week on October 19th, Thursday, the same time, the same Zoom link. We'll have Professor Karen Polizzi at Imperial College London and Dr. Lin Su at University of Cambridge. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with today's speakers. I will promote you to panelists who could speak and show your handsome and pretty faces if you wish. Thanks. I stop recording. Thank you.